In this section, we're going to talk about gram-negative proteobacteria. Proteobacteria are some of the most different of any of the groups. They have different shapes, different sizes, different nutritional requirements. A lot of them are pathogenic, some of them are phototrophic, on and on and on and on. So, why do we put them all together? Well, because based on 16S, our RNA genes, they are closely related, and they're all gram-negative, which means they have a thin peptidoglycan cell wall, on the outside of which they have an outer membrane, in which you have lipopolysaccharide, or LPS. So, here's how I think of it. Think of it as you have a founding family, and the brothers and sisters go off, get married, and after 3 or 4 generations, you've got a group of related people that are all very different. They look different, they have different professions, and yet they can all trace their ancestry back to the original couple. So that's kind of like how the proteobacteria are. And one way to remember these guys is that proteo refers to many shapes. That's the Greek for that. There are 5 sections. There are the alpha proteobacteria, the beta proteobacteria, the gamma proteobacteria, which is the biggest group, the delta proteobacteria, and the epsilon proteobacteria. So, let's go ahead and take a look at them. From the learning objectives, there's a couple of things that you need to remember about the alpha proteobacteria. And the first is that some of them have this unique structure, which is called a prostachia. And this increases their surface area so that they can absorb more. Also, this serves as a way to attach to surfaces and other cells. So, that's enough about the prostachia. Next up, 2 varieties of alpha bacteria that benefit mankind. And these are all nitrogen-fixing bacteria. They take the atmospheric nitrogen and they convert it into ammonia or other organic molecules in which the nitrogen is accessible. So let's talk about rhizobium, which is found in legumes, because that's the one that you're probably most familiar with. They're common soil bacteria, but legumes, like these soybean plants or peas or beans other than green beans, alfalfa, send out signals and saying, hey, I'm here and I would like to host you. The bacteria move through the soil and enter the roots of the legumes, and this plant forms these nodules. And they protect the rhizobium from atmospheric oxygen. So, just like cyanobacteria, the enzyme in this genus of bacterium is killed by oxygen. So, they're able to fix nitrogen. The plant provides the sugars through photosynthesis, and the bacteria provides the nitrogen that the plant needs, and there's extra that leaches into the soil. So that's why we rotate legumes with our other crops that don't form these associations. So does this relationship sound similar to the cyanobacteria? It does, doesn't it? Very interesting. Now the azospirillum forms a similar relationship with some tropical grasses. Rhizobium is found in temperate areas. Azospirillum performs the same ecological function in tropical areas. Now on to the pathogens. Rickettsia is an alpha proteobacteria, but it doesn't fix nitrogen. In fact, it has lost the ability to use glucose. It doesn't undergo glycolysis. But to survive, it still needs the intermediates of the Krebs cycle. Well, if you don't have glycolysis, you really can't do the Krebs cycle. So Rickettsia are obligate intracellular parasites. In other words, they live inside of other cells so that they can get those Krebs cycle intermediates. Now, there are several species of Rickettsia that cause human disease, and they're transmitted by ticks, like Rocky Mountain spotted fever, which interestingly enough is not found in the Rocky Mountains very much anymore. Most of the time it's found east of the Rocky Mountains, but nevertheless. Or lice, which is a relative of ticks that transfer typhus, which has been a bane of armies throughout recorded history. So we got the good guys and we got the bad guys. To meet the objectives for this topic, we need to talk about 3 beneficial beta proteobacteria, and we need to talk about 3 non-beneficial beta proteobacteria. Just to change things up, let's talk about the pathogens first. So we have 2 genera, Bordetella and Neisseria. Bordetella pertussis is the causative agent of whooping cough, pertussis, which is still a serious childhood disease, but we have a good vaccine for it. Neisseria has a tendency to form diplococci, and this is characteristic for the genus. So Neisseria meningitidis causes meningitis. It's one of the causative agents of meningitis. And Neisseria gonorrhoeae, well, that causes gonorrhea, which is a sexually transmitted disease, or STD, which has been on the rise recently. Unfortunately, we do not have vaccines for either one. Now, on to the beneficial gram-negative beta proteobacteria. Theobacillus is a bacterium that is important in the cycling of sulfur in the environment. It oxidizes either hydrogen sulfide or sulfur, depending upon the species, into sulfates. And other organisms can use sulfates to incorporate sulfur into their amino acids. So they're important in the environment. Nitrosomonas is important in nitrogen fixing. It doesn't live in association with any plants, but it does fix nitrogen and releases ammonia into the environment so that other bacteria and other organisms can use the ammonia to incorporate nitrogen into their proteins and their nucleic acids. And the last one, zooglia, forms masses or flocks of organic matter and bacteria in wastewater treatment tanks, and they have a tendency to sink out of suspension, aiding in the purification process. So we are very glad to have zooglia helping us with that process. Next, on to the gram-negative Grama proteobacteria, which, as I've mentioned before, is the biggest group of proteobacteria. Now, you may have heard of several of these. You've heard of E. coli. We use that in the lab. It is a common inhabitant of your gut. But strain 0157H7 is the kind that causes the hemorrhagic uremic disorder. That's the one that if you get it in the uncooked meat, it can cause bloody diarrhea, shut down your kidneys. That's why we cook our meat very well. They've also been found to contaminate everything from alfalfa sprouts to spinach to tomatoes to cucumbers. Anything that can be contaminated with cow manure can spread 0157H7, which is usually found in the guts of cattle. Salmonella! Salmonella causes food poisoning as well, and this is one of the few microbiology jokes that I have that is actually funny. Shigella you may have heard of. It's related to E. coli, but it causes bloody diarrhea or dysentery. Yersinia pestis causes the plague. You want to watch out for Yersinia. And let's get rid of my cartoon there. Vibrio cholera causes cholera. And you can see it's kind of a jelly bean shape here. Sometimes it's more comma shaped. That's where the Vibrio comes from. Then there's Pseudomonas. Pseudomonas aeruginosa causes some opportunistic diseases. They have a tendency to show up in ear infections, and they're very hard to treat with antibiotics for whatever reason. They also like to grow in quaternary ammonium chlorides and soaps and detergents. They'll eat just about anything. And they're unique in that they divide longwise rather than through the short ways, like most other bacteria. So you can see from this survey that we've got all sorts of different kinds of gamma proteobacteria, including some that I haven't talked about, like purple sulfur bacteria. We've got some obligate intracellular parasites, and on and on and on. But these are the ones that are clinically important. On to the delta proteobacteria. We're going to talk about Delvibrio because it's fun. It preys upon other bacteria, and especially gram-negative bacteria, so they eat their cousins. They attach, like you can see in this micrograph, and they spin and drill their way into the periplasmic space between the cell wall and the cytoplasmic membrane, and then they get really, really long, unlike most bacteria that start dividing when they are twice as long as they used to be. Once they get really, really long, they do a whole bunch of divisions. They've finished eating the cell. They lyse the cell membrane, they lyse the cell wall, and go out and prey upon other bacteria. Kind of cool. Creepy, but cool. Another delta proteobacteria are myxobacteria. And you're probably saying, we've got too many MY bacteria names. Why couldn't they pick something else? Well, these guys used to be in with the slime molds because they behave in a similar way, but they're prokaryotes. They're not molds. They got kicked out of the molds. And eventually the slime molds got kicked out too, so they're not molds either. But we maintain the scientific terminology for mold in the title. So these guys alternate between living as individual cells that live in the soil, they glide along, eating bacteria and other types of things. When food gets scarce, they start emitting signals that cause them to gather, and they form this fungi-looking fruiting body. And the cells that are lucky enough to end up in the top turn into kind of a spore, and they blow in the wind, hopefully landing in a different spot where they can become vegetative again and proceed to hunt for more food. Now, these aren't endospores. They're not as resistant as endospores, but they are a somewhat resistant type of cell. Now, the cells that end up in the stalk sacrifice themselves for the cells that end up here. They end up dying. And you can imagine going from this to a multicellular organism. So they're almost a multicellular prokaryote, except for they have this free single living stage. But it's very interesting, and scientists study the signals that cause the individual cells to gather and to differentiate into the cells that form the stalk and the cells that form the spores up at the top.